This is the Master Brewers Podcast, brought to you by the Master Brewers Association of the Americas, a volunteer organization dedicated to continually improving the products and processes of our membership since 1887.
Let's go!
Master Brewers brings you interviews with the industry's best and brightest in brewing science, technology, and operations.
Calm down, I'm moving too fast.
This Master Brewers podcast is proudly sponsored by Hopsteiner, a global leader in the hop industry focused on quality, sustainability, and innovation in new hop varieties and hop products. Contact our brewery sales team to provide you with the hop-related tools you need to craft your next great beer. For more information, visit hopsteiner.com.
Additional support provided by
Get to know Proximity Malt. We malt superior, European-style, low-protein varieties grown close to home in Delaware and Colorado. Domestically grown, precisely malted to style. With our team of seasoned experts and 2 brand-new malt houses, try what's really new in malt. Check us out at www.proximitymalt.com.
Additional support provided by
The secret to quality fermentation is White Labs. Core strains are made weekly and most can ship out the next day. Order through the White Labs app or yeastman.com. Visit whitelabs.com/whywhitelabs for a new customer special offer. That's whitelabs.com/WHY Whitelabs.
Okay, I'll start. My name is Maitreya Dunham. I'm a professor at University of Washington in the Genome Sciences Department.
And my name is Tom Schmidlin, and I'm owner and head brewer of Postdoc Brewing in Redmond, Washington.
This week on the show, genome sequencing of cereal repitches.
We observed a couple of different key genome changes. So the one of them is that about 40% of the population gained an extra chromosome.
Just about everyone in the brewing industry will recommend not repitching most yeast strains more than 10 times. There's really nothing magic about the number 10, but let's talk about why we want to limit the number of repitches. What's the math look like?
So one reason to limit the number of repitches is because eventually evolution is going to cause trouble for you. And that's because there are so many yeast cells in a fermenter, 10 to the 14th, which is one with 14 zeros after it, that there are lots of possibilities for mutations to happen. And if a mutation happens that makes that particular yeast cell better at growing or surviving in the beer, then it's going to start to take over the fermenter.
Tell us more about what those mutations might look like.
Yeah, so the changes can be all sorts of really cool things. So that's one thing my lab focuses on, actually, is how the genome evolves. And we got interested in beer because it's such an interesting environment in which yeast can evolve. And the types of mutations are the same as any type of evolving system. There are individual spelling changes in the genome where you can change a single base pair. They are whole chromosome changes where you can come up with an extra chromosome or lose a chromosome. And you can also swap chromosome segments between each other. And these can have really profound consequences for the metabolism of the yeast strain. They can start making different quantities of different metabolites, or they can grow differently, or they can have different stress tolerances, in part because of these mutations.
Okay, and just to be clear, because a lot of brewers misuse this term, generations and repitches are not the same thing. There are multiple generations within any given fermentation.
Yeah, that's right. So we tried to look this up, and it turns out the data are not super solid on how many cell divisions are occurring in each fermenter, but it seems to be on the order of about 3 population doublings in a fermentation. And so the repitch going to the next fermenter will include more cell divisions than just the fermenters used.
Yeah, I'd say that number's about right, especially for the the strain in question here that you did a lot of work with in giving kind of sort of normal fermentation conditions and whatnot. But yeah, I think that number probably varies a lot from one situation to another. Now, your lab studies these mutations to learn about the evolution of yeast during cereal repitching. Why is that important?
So evolution is really just fundamentally interesting. It's this basic biological process. And so one of the things my lab wants to work on is how it happens and how mutations can affect traits of cells. But it also has a lot of implications for both industrial applications like beer, but also, a lot of biopharmaceutical products are made in fermenters these days by yeast. And so they have some of the same problems where, you've built this nice pathway in yeast and it's making some small molecule for you, but evolution manages to screw it up for you and you don't get the yields you want. The other reasons to study evolution are because there are some big public health problems that are where evolution is really important. So one of these is antibiotic resistance, for example, like microbes evolving the capacity to be resistant to a drug that we need in order to fight those infections. And another one is cancer. So cancer cells are actually your own cells evolving to grow aberrantly in your body. And all of these processes, the fundamentals are the same. It's all mutation and growth and competition, but the specific types of environments that they're in can differ. And so that's one reason why we've been studying this both in the lab and also in the brewery, is to try to understand how different environments matter.
I've always thought that beer is going to somehow cure cancer, so you're giving me more hope now. Okay. You've said that brewing strains are monsters with crazy chromosomes. Explain what you mean by that to those of us who aren't geneticists.
Sure. So if you think about your own genome, you have 23 chromosomes, and you have 2 copies of each of those chromosomes. You got one from your mom and one from your dad, right? However, and that's also true of most yeast strains. However, the beer strains have very interesting and crazy genomes. They don't just have 2 copies of each chromosome. They have sometimes 3 copies, sometimes 5 copies, most of the time 4 copies. Some chromosomes aren't entire chromosomes or partial chromosome fragments that have gotten hooked up to other regions of the genome. So there are these really interesting genomes that are really different from what a normal yeast cell looks like. And so some of those changes in the genome are no doubt responsible for some of the great traits that are already present in all of these neat brewing strains. And I should say that that's not entirely true. Like some strains are near to 4 copies of each chromosome, but other strains are closer to 2 copies of each chromosome. It depends on the history of that particular strain.
So take us to the beginning of this work. How did you get started?
So I saw a poster for a talk that Tom was giving at University of Washington. He's an alum and had been signed up to do a career seminar for what could you do with your awesome science degree. And I think I emailed out of the blue to ask if you happened to be around to meet, because we were just starting to do some beer genomics work. And we met, and you told me about your cereal repitching, and I was like, please take samples for me. Oh my God, that sounds amazing. Am I remembering it correctly?
You have a better memory than I do.
All right, so let's hear about how this experiment sort of came to life. How did you set this whole thing up?
Serial repitching is just something that we've always done. We've only been around for 4 years, but from the beginning, we harvest our yeast and reuse it. And I've never been afraid to, just keep going with it. I know that there's some rules of thumb in terms of, how many generations you should go out in terms of repitching, but
rules are meant to be broken, right?
Well, if it's still tasting good, I didn't see the reason to throw it away. And speaking with some other people in the industry, I know, I'm not mentioning any names, but I know of at least 2 other breweries in the region that go out far, far further than we do in terms of the number of batches they'll get out of a single pitch of yeast.
Oh, Tom, you got to tell me what their names are so that I can go get samples from them too.
I'll have to get the permission before I do that. Yeah.
Yeah. So what we wanted to do was start from the beginning. So we asked Tom and his crew to begin collecting samples for us the next time they started from a fresh batch of yeast. So they ordered a new batch of yeast from Yeast, and then collected a sample for us from that and from the prop up, and then started collecting essentially every single time they used the yeast, they collected a sample for us. And so luckily, one of Tom's folks lives pretty close to the university, and so was able to drive by yeast samples on a regular basis for us. And we accumulated them all in our freezer until he decided that the time series was over. And as he said, that was because the taste started, not being what he was looking for. So once we had all these samples in our freezer, we chose a few of them from about every 5 to 10 repitches. And we made DNA from the mixture, and we used DNA sequencing to sequence the entire genome of the yeast and of the entire mixture. So what we have at most of these time points is a mixed population of a variety of things going on, and we can watch as new mutations kind of increase in their frequency throughout the time course. The important thing was that we had the time zero, that first sample that we got from the yeast company, so that we had something to compare it to, so we could tell what was new and what pre-existed.
Nice. So in your first experiment, you ended up repitching Chico yeast 29 times. So that's something like more than 80 generations. Tell us what happened. What did you observe?
Well, first of all, we observed a couple of different key genome changes. So the one of them is that about 40% of the population gained an extra chromosome. So yeast have 16 chromosomes, and a portion of the culture now has 4 copies instead of 3 copies of chromosome 5. So that was pretty easy to see. And then the second one was a little bit more subtle. And so another 40%, and this is separate, they aren't the same clone. So one group of 40% of yeast have this extra chromosome, and then a different 40% of the final pitch of the yeast have a rearrangement where it doesn't change the number of chromosomes, but it changes essentially which copy they have. So if you think about the yeast as having, 2 chromosome copies from yeast mom and yeast dad, what's happened is that they started out with 3 copies in this case from mom and 1 copy from dad, which is a little weird, but go with me. And now at the end, they have 2 copies from mom and 2 copies from dad. So that's a pretty subtle change, but what we think is important about it is that there's a mutation carried on one of those chromosome arms that pre-existed in the strain, and we think that this rearrangement is getting rid of it. We don't yet know the flavor implications of that. We're in the process of engineering an ale strain to carry just that mutation and no other mutations so that we can test it directly. And then the other types of changes we saw were not whole chromosomes or whole chromosome arms changing copies, but instead were more surgical mutations where just a single base pair might change. And we saw quite a lot of those. Only a few of them made it to relatively high frequency in the population. One of those was in a gene called SCIP2, which interestingly regulates hydrogen sulfide production. So this was one of our potential culprits for any kind of off flavors that might have been developing. Although, again, we haven't followed that up to prove it or not.
Once you identify a mutation via sequencing, how do you then figure out the effect of that mutation? How do you determine, okay, this mutation affects flocculation or ethanol tolerance or VDK production or something else?
Oh, that's like the $1 million question. That is what all geneticists kind of obsess over these days. So it's kind of called the genotype to phenotype problem. If you know the genotype of something, how can you figure out like what changes it's going to make to the traits of the organism? And one great thing about working with yeast is that it's been studied, like back to Pasteur, right? So there's a ton known about yeast. It was one of the first genomes sequenced, and people have been working hard on it for decades. So a lot of the metabolism is really well understood. And so when we find a mutation mutation in a gene, we can make some guesses about how it might affect the physiology of the cell. But to prove it, what we really need to do is engineer that mutation and only that mutation into a strain, and then compare it to a control strain that doesn't have that mutation. And then we know the only difference is this one change that we made, and then we can do the bake-off, right? We can have them grow in different conditions. We can measure the metabolites that they create and see if we can figure out what difference does that mutation actually make. Or was it just there by random chance? So one thing that we found really interesting with these the strains that had been repitched so long is that they didn't just have one mutation. They often had several. So, they would have the extra chromosome copy, but then in addition to that, they would have one of these point mutations. And so sometimes there's some ambiguity there. Like we might see that the strain has a difference when we compare it against the initial strain, but we don't know yet which of the mutations causes it. And even to make things worse, for these whole chromosome changes, like there are hundreds of genes on chromosome 5. So which one of those genes is the one responsible? Or maybe it's not just one gene, maybe it's a combination of them. It makes things really complicated and really interesting.
But it sounds like you're telling me you're in a pretty good position to solve this riddle and figure this all out for the world, right?
We're trying. You might have heard about CRISPR. We've got a CRISPR experiment going right now, and one of the starting with the Chico strain to try to engineer some of these changes in and see. Because the other thing is that evolution of these strains doesn't necessarily have to be bad. It can also create strains that have interesting and good new properties. So, for example, these strains, when we grew them in high ethanol concentrations, so 7.5%, that kind of impaired the growth a little bit of the initial strain, but the repitched strains actually grew better in that condition. So it could be that these strains are going to be useful because they're more tolerant of some of the brewing conditions in Tom's specific brewery. They haven't been, kind of optimized for his specific brewery. They've been collected for their general brewing characteristics. So maybe we'll evolve something cool that Tom will want to use later.
Yeah.
Nice. Yeah, you have to check out, we did an episode last summer with Charles Denby, who was using CRISPR to make hoppy beer without using hops.
Yeah.
So
I love that study.
Yeah, it's a pretty good one. Coming up.
And we've collected a whole bunch of different ones, and we're trying to kind of understand what happened in the past as these strains got passed around, and then also what happens in the future as they get repitched in individual breweries.
I'm John Bryce, and you're listening to the Master Brewers Podcast from the Master Brewers Association of the Americas. Support for this podcast is brought to you by
ABS Commercial is a full-service brewery and parts outfitter. From our Raleigh headquarters to our Denver office, we proudly offer brew houses and fermenters from 3 barrels and up, yeast brinks, boilers, kegs, chillers, tri-clamp, and other stainless parts, all with the quickest delivery and lead times in the industry. Learn more at abs-commercial.com or call 877-BREW-ABS. ABS Commercial. We are brewers.
Additional support provided by
Bring the world to your brewhouse with BSG's diverse selection of ingredients and services. Our dedicated customer service team and industry experience provides you with the assistance you need every step of the way. Make BSG your supplier of choice with products essential to making great artisanal beverages so you can stay focused on your craft. Visit us at bsgcraftbrewing.com or contact us at 1-800-374-2739.
Here's what's coming up on the Master Brewers calendar. District St. Paul, Minneapolis meets May 16th at the Star Keller in New Ulm. District St. Louis is at Old Bakery Brewery May 16th. District New England meets in Newport May 17th and 18th. The District Southeast Spring Meeting and 4th Annual Crawfish Boil is May 17th and 18th in Tampa. District Rocky Mountain meets at Red Truck Beer Company in Fort Collins May 21st. And District Northern Illinois meets at Half Acre Beer May 31st. Don't miss the CO2 monitoring and the brewery and brewpub webinar June 13th. District Philly's annual golf outing is June 14th. And District Midwest meets at Wolf's Ridge Brewing in Columbus June 29th. It's time to start making plans for the 2019 Master Brewers Conference. If you can only make it to one conference in 2019, this should be it. We're really mixing things up this time and heading to the Calgary Convention Center to see how Alberta celebrates Halloween. Check out the full count of events at mbaa.com for more details or to find a district meeting near you. Now back to the show. I guess let's hear more about the mutants that were uncovered during this study. What kind of superpowers did they have?
So once we had these mutants that we'd discovered, we wanted to see if we could find some reason that they took over in the brewery. Because going from undetectable at the beginning to 40% of the population when you have these huge fermenters and only a small number of generations is actually really amazing. So they have to be really good at something, and we wanted to know what they were really good at. So we started trying to think of what are all of the environments and stresses that the yeast have to undergo, and could we start testing those individually in the lab and seeing if we could figure out what kind of new superpowers these strains had. So in order to do that, what we did was we isolated individual clones that had the mutations. So before on the sequencing, we'd done this whole population that was all mixed together. So in this case, what we did was we pulled out single cells and made sure they had some of the mutations we were interested in, and then started growing them in the lab. And so a few of the things we tried were, we thought maybe they were more cold tolerant because Tom puts these strains in the cold room between batches, so we thought maybe they were having some advantage while they were hanging out waiting for their next fermentation. We didn't find any traits related to cold tolerance, although we might look a little bit harder for that later. Another one that was a good possibility was flocculation. How they settle into the cone would determine who gets brought over to the next batch. And so there are a few laboratory tests, and this is actually a trait that my lab had worked on before. So we already had some of these protocols already established in the lab. So we compared them in terms of their flocculation properties. Again, we didn't find any differences there, but of course we don't have a giant fermenter to try this out in. So it is still possible that they have some sort of difference in Tom's fermenters that we weren't able to capture in the lab. It could also be that they just grow faster. So to do that, we call it science beer, where we've figured out a way to make sterile wort in the lab that's pretty reproducible so that we can test things over and over again. And sure enough, the strains collected from the end of the repitching time series did grow faster than the original strain did. So we think that could be one component of why they're able to take over by the end. The other trait that we did find a difference in is ethanol resistance. And so if we grow the strains in higher concentrations of ethanol, then we found that the final strains with the mutations did grow a little bit better than the starting strain did in about 7.5% ethanol. They don't make more ethanol. We tested that by actually making beer with the strains and then sending it off to white labs for analysis. And the actual concentration of ethanol that they produced was about the same. Tom also made beer with our strains that we sent him. And so we tested those as well and got some of the same results.
Nice. Okay, well, one thing I wanted to bring up is that the cereal repitches you studied took place in a commercial brewery across a dozen different beer styles. So that yeast was repitched into a different environment each fermentation cycle. I'm sure you're curious as to what extent the varying stresses of those different environments affect mutation.
Oh, that's a really great point. If we could find breweries that repitch through the same beer over and over and over again instead of kind of being an all-purpose strain, then we could try to do that experiment where we could maybe find an all IPA route and an all stout route and see how they change in different ways. That's easier said than done. If anyone out there listening knows of breweries like this, please put me in touch with them because we would love to see if they'll collect samples for us.
Tom, it was actually quite a coincidence coincidence because about an hour after I emailed you an invitation to appear on the show, a post from Christopher Large, who's a PhD student in your lab, came through on Ask the Brewmasters at community.mbaa.com, and he was soliciting commercial brewers to participate in your research. So are you still, it sounds like you're still looking for more breweries to get involved. Do you want me to post a link to Christopher's post in the show notes here?
That would be fantastic. And we have gotten great responses from several other breweries. And so we're in the process of beginning to collect samples from them and see what people have for us. I've been really thrilled at how willing people are to share things with us. And we have two asks. One is for other breweries that are doing serial repitching. But the other is that one of the interesting things that we noticed about the strains was that it seems like a pre-existing mutation in the strain went away, and not all Chico strains turn out to have that mutation. So now we've gotten really interested in how did all the Chico strains evolve as they got passed around from different breweries. So we're trying to collect everybody's house version of the Chico strain, and we've collected a whole bunch of different ones, and we're trying to kind of understand what happened in the past as these strains got passed around, and then also what happens in the future as they get repitched in individual breweries.
That's cool. I mean, I can attest without, doing any kind of, research on it, but just having brewed beer in a lot of different breweries over a lot of different years and using American ale yeast, Chico strains from lots of different suppliers, they're not all the same for sure.
Oh, absolutely not.
Yeah.
So we already have genome sequence from Yeast 1056 and White Labs 001, and they have some of these same types of differences that we saw during the repitching. They have extra chromosomes. They have chromosome rearrangements on some of the arms. So they're already reasonably different from each other in interesting ways. And, there are a few reports of people doing head-to-head comparisons and not seeing a lot of flavor differences from these, but there are some reports of some behavioral differences. So I don't know. So one reason that we wanted to look at the repitching is because it's a smaller number of mutations that happen over that short period of time, whereas there's quite a large number of mutations that have happened between 1056 and 001. So figuring out which one of those mutations or multiple of those mutations are responsible for anything is really hard.
That's cool. So that post that Christopher put out was back in October. So I'm sure you've repeated that study, quite a bit since then. Did you get the same mutations when you've repeated the study, or has it been for pretty different each time?
So Tom is currently in the middle of our repeat. It's going really well, and it's a particularly long time course, which is really exciting. So maybe Tom could talk more about that.
Yeah, let's hear about it.
Well, obviously, I don't know any of the results. We're just, doing the same thing we did before, repitching from batch to batch to batch as we go. It takes us a pretty long time to get out to 30 repitches because we're, dividing as we go. And also this is not the only yeast strain that we use. So, we have many other batches going with different yeasts, and we've actually started to use the A38 strain from Imperial Organic Yeast quite a bit. And so that is also, as we're repitching those, we're sending samples to Maitreya's lab. It's basically a repeat of the previous experiment in terms of variety of styles and pitched again and again. We just, don't have all the results yet. We're still using it.
Cool. How far along are you?
Let me check here real quick.
He's like, I don't know. I just put yeast in a jar and sent it to her.
We, the last sample that we delivered was repitch 29. And so that's obviously going to lag behind where we are. So that was in the bond. So we're probably at 31 at this point.
Nice.
Wow. I have to say, one of the great things about working with Tom is that he does keep remarkably good notes. And also, I have the feeling he has pretty good technique with his background in science. And so that could be one of the secrets to his success with this long-term repitching. Because we talked about how mutations are one thing to worry about when you repitch. But of course, the other things to worry about are that you introduce contamination or that the yeast physiology starts to suffer.
That's right. And I guess, Tom, you're in good shape because you got someone who can tell you if you ever have any cross-contamination.
Yeah, exactly. Well, we test our tanks every week anyway.
Right. You can't hide from the sequencer, though. That's true.
That's right. Cool. Well, this is very interesting stuff. I didn't ask that many questions. Is there other stuff that either of you want to talk about that I didn't ask about?
Oh, let's see. Did you want to talk a little bit about the beers that Tom made that we brought to HomebrewCon for tasting?
Yeah, why not?
That was kind of fun. So Tom actually, so we made science beer with the strain.
Is that where you gave this? Is that where this presentation is from? Or
yes,
okay.
That's right. It's from.
I don't even know where it's from. Somebody sent it to me. He was like, "Yeah, you ought to do this."
Funny.
And I was like, "Oh, yeah."
Yeah, no, I didn't know that they kept the slides like this and posted them. I knew they were posting the talk online, but yeah. I'm glad I did a good job on the slides. It's kind of funny. There's some things missing. I'm looking through it.
It's the internet. Everything lives forever. You have to be careful.
Yeah, right. Well, I'm just wondering. So far, everything has held up from a few months ago, which is good. But let's see. So one thing that was great fun was that Tom actually made beer with the two mutant strains that we isolated, with one with the extra chromosome 5 and one with the chromosome rearrangement, and also with the starting strain. And then we brought them to HomebrewCon and had a whole room full of homebrewers do sensory analysis for us, which is great. And so they were all like instantly, I had 200 new collaborators, right? And we are still in the process of analyzing all of that data, but does look like we saw some differences that were measurable by all of the brewer tasters. So we got some differences on the ester score and the sweetness score and the hop score. One of the beers in particular scored a little bit higher than the other two in all three of those areas. And we are, again, not entirely sure what's causing those differences, but they do agree to some degree with some of the metabolite analysis that we had done, where we saw different concentrations of some of these small metabolites that can change the flavor. And Tom did some tasting too and said he could tell the difference, so I trust his palate.
Yeah, when we did the taste test at the brewery before the Homebrewers Conference, we there were clear differences between them, and we did those tastings blind. But it was very obvious the different strains, and it was, I mean, they were so distinct that when we were at the Homebrewers Conference and I tasted them during the talk, I could immediately pick out which was which. They were very different from each other. But the thing to keep in mind with those is that the final yeast that we had where we decided to end the time course was 40%, let's say one strain, 40% another strain, and then 20% unknown, really. Could be a mix of other strains,
right?
But certainly not a monoculture. But when we made these beers, we made one beer with one of the 40%, and one beer with another of the 40%, and then nothing with that 20% of who knows what that is. So really we're isolating the different mutations and brewing beer with them, and then tasting them to see what the differences are. So those differences, when it came time to, when we're talking about the beers that we were making with that, right before the time course ended, maybe those differences cancel each other out. It's possible that conditions here are not quite the same as the beers that we made for the conference, were fermented in carboys, homebrew style. I mean, we made a 15 barrel batch of the base beer because, it was a good style to test this out with, and we were going to, brew a batch of pale ale. Anyway, so we took this pale ale wort and fermented it with these different strains, but they, like I said, they were in carboys. They weren't as controlled as they are in our normal fermenters. We're not able to, do a reliable diacetyl rest with that equipment as compared to our normal equipment. And so, there's some things that I think came across in those, let's call them homebrewed batches of beer, that you wouldn't see in the beers that we normally produce.
We've heard a lot about sort of the population that you started with and the mutants that we ended up with at the end of a whole bunch of serial repitching. But what happened in the middle?
So in addition to the final time point, we also sequenced some of the time points along the way, and that let us see what was happening in between, going from the mutants being undetectable to being at about 40% of the population. So we were able to start detecting the presence of these mutants by about 10 to 15 repitches. So what's interesting is that mostly agrees with that kind of rule of thumb, right? That 10 repitches is about what you can get away with. And that seems to be kind of when these things start to have enough presence in the population to make themselves known. We are looking to see in the replicate if we get that same result again, or if it ends up being a little earlier or later.
I assume you didn't sequence, every cycle just because that would have been way too much work, right?
Yeah, we didn't know when things might happen. And in fact, it's still pretty amazing to me that we have so much of the population that's a mutant by the end, because it really is a pretty small number of generations in the grand scheme of things. Like in my lab, when we do laboratory evolution experiments, we will often do hundreds of generations. So although we have much, much smaller cultures than the fermenters.
That's cool. And I suppose, if you end up finding that there's a really interesting mutation, you would be able to drill down further and go back in sequence to figure out exactly when that happened, right?
Absolutely. So we still have all the samples in the freezer. It's kind of like a little fossil record of all of Tom's brewing activity for that 6 months. So that's what we kind of started with, was the sparse sampling first. And then once we figured out where things were happening, then we could go and do more samples in that area if we needed to.
Yeah.
Yeah, you wouldn't need to do whole genome sequencing on those just to find, if you're just trying to track back to see where a single mutation happened.
Right. Okay.
That's right. There are also cheaper methods to go and track down individual mutations. So like, let's say we found a single base change that we got really interested in, we could use other methods of detecting that are actually a lot more sensitive than whole genome sequencing, where you could figure out, like, is this at, less than 1% population frequency? Whereas with the whole genome sequencing, we're really looking more on the order of 10%.
That was Maitreya Dunham and Tom Schmidlin here on the Master Brewers podcast. Don't forget to check out Chris Large's post on Ask the Brewmasters if you'd like to participate in this study. I'll post a link in the show notes, or you can just type large into the search bar at community.mbaa.com.
See, I'm glad you brought up Chris, because I do want to emphasize that Chris Large in my lab is the one who's done all of the sequencing work on this. And Noah Hansen, a tech in my lab who got his start in microbiology as a home brewer, is the one who came up with our science beer recipe and has also been doing a lot of the helping with the sequencing work on this. So I definitely want to make sure both of them get acknowledged.
It's time to start making plans for the 2019 Master Brewers Conference. If you can only make it to one conference in 2019, this should be it. We're really mixing things up this time and heading to the Calgary Convention Center to see how Alberta celebrates Halloween. You can find all the details on the meetings tab at mbaa.com. Did you know that Master Brewers now has a mobile app? TQ articles, podcasts, webinars, Ask the Brewmasters, and more, all in the same place. Search Master Brewers in the App Store to download it now.
Losing too much, and then I'm heading out to any other place. My fist full of courage, my heart full of rage. I can't get stuck, I can't be